Hello, my name is Professor Barrett from the University of Dundee in Scotland. Today I'm going to be talking to you about how to design your study in an IVF clinic and a laboratory. I don't believe I have any significant conflicts of interest. I'm a fully employed member of the University of Dundee, which pays 100% of my salary. I have a research, teaching and administrative post in the university. These opinions for the presentation are entirely my own. They don't reflect those of a particular journal I'm associated with, or in fact the University of Dundee. Somebody else giving this lecture would give you a completely different viewpoint and probably very many different examples. I've been given a small honorarium to help prepare and present these lectures to you today. So let's get down to the lecture plan. The first thing to say is that studies are actually vital. Despite our biases, I guess, we don't know the answers to everything. So we need to be able to do a study to try and determine, to reveal what the likely answers are to our questions. There are a plethora of resources available to you to actually do any type of analysis and study in this area. They can be from journals, for example, they can be from YouTube videos, they can be from academic resources, they can be from colleagues. Whatever you get information about it and how to design and execute studies, you need to be really well prepared before you start your study. I call this the 6 Ps, which is prior, planning and preparation, prevent, poor performance. I'll go through the basics for a study. I will also talk about the studies with ART examples, so we can try and get a real flavor from the literature about what was happening and examples from studies. I will talk about specific challenges within an ART clinic and laboratory to doing, trying to do studies, experimental studies, clinical studies, because I think it's important that although there are general points where we need to actually get sorted on how to do studies, there are specific challenges that we do face, and we do need to recognize those and be able to deal with them. And of course, there is, what do you do next? So I've got some experience of writing papers. I've written a number of primary data papers and reviews. These have been published in general journals, such as The Lancet as an example, and in a number of reproductive medicine journals, such as Fertility and Sterility, Human Reproduction, and MHR. I've also been on editorial boards of a number of journals, and I've been part of workshops that have been dealing with study design, experiments, how to get your papers published, how to do a study, etc. So I've got some experience in the arena. I think the first thing to say is that studies are absolutely critically important and necessary to society. There is no getting away from the importance of this. And let's take a recent example. So this week, for example, Pfizer, in collaboration, have actually come out with a study results related to a COVID vaccine, which has been a major breakthrough, potentially, of course, in our whole lifestyle and how we deal with COVID-19. So actually designing a study testing this vaccine has been really, really important. But of course, all of the experiments prior to that to try and get to the stage of which vaccine to use, what's the mechanism of that vaccine, which type do we use, which design of trial do we use, etc., has been a massive scientific effort. But to me, it shows the power of science in trying to actually deal with particularly global issues that we face. So one of the challenges that people face when they're thinking about studies to do and experiments to assess is that actually they say, well, don't we know most things, particularly in reproductive medicine? You know, IVF works, for example. And the truth of the matter is that actually we have a lot of ignorance about many processes involved in reproductive medicine. The example I use here just comes from IMSI. So IMSI has been around for some time, and this is a Cochrane review, very recently published in 2020. And we can see here that the conclusions of the study were very much, after the analysis of all of the data, they found very low quality evidence that IMSI may increase the chances of a clinical pregnancy, which actually means that they are uncertain yet about any real difference. And this could apply to a number of things in reproductive medicine. So actually, we don't know the answers to many questions, and even though we think we do. So that is a very important lesson. There's plenty to be done in reproductive medicine, and we actually need to get on with these types of studies. Now, it is very important, particularly with regard to that subject, to give a health warning. Science and biology, reproductive medicine is actually complex. It isn't binary, it isn't simple when you're trying to address experimental questions and clinical questions. And I think it's fundamentally important when you start to realize that actually biology isn't simple. It is complex. And therefore, when you're trying to understand how a system works or why it works, you're unlikely to be able to get a clear answer just by a cursory investigation. So you need to understand the health warning that science actually is complex. And what we think we understand and what we think we know, they can be quite challenging at times. So let's look at the very basics of doing a study. And this could apply to pretty well much any study you're doing. It could be looking at how to make a cup of tea, the extreme example, but it could be to anything, how to build fences, whatever. But this is the basic of a study that I think is quite important to look at. First of all, and this is just completely critical, you need incredible enthusiasm and motivation to do a study. Let me give you an example. So David Miller started in probably 2009, 2010 to actually look at Hab Select pixie dishes as a potential study in reproductive medicine to see whether they actually worked or not. He then needed to get the money, design the trial. Once he got the money and the trial was designed, he had to execute the trial. And that took obviously quite a lot of work. And then, of course, you got to publish the paper. And the paper wasn't published till 2019. So pretty well much 10 years of the author's life would have been occupied by designing, putting together, funding, and driving that study to get an answer. Now, that's an extreme, of course. But I think it's important to understand that actually studies can take some time to do, particularly that the answer isn't very simple. So you need a lot of motivation and enthusiasm and tenacity, in fact. You must also be able to think. And what I mean by this is not, Nobel Prize winning thinking necessarily. What I mean is you can't be asleep. And I've known many, many people doing studies who actually, they're awake, they can talk to you, make a cup of tea and stuff, but they're actually asleep intellectually. And that doesn't help you at all when you're trying to do a study. So I think that's point 2. Point 3, you've got to consider what question you're going to ask really carefully, because this is critical to what you want to do. Because if you ask the wrong question related to what you're trying to find out, you're going to go completely along the wrong road. You've then got an intelligence gather. And then once you've intelligence gathered, you can start to formulate a working hypothesis. I always find it very useful to try and think, well, what's my likely answer? One answer would excite me. And if I do this experiment, what are the options to actually come out as an answer? And you can play with that on a piece of paper and a pencil, for example. I think then you've got to consider your different study designs. And there are lots of these different study designs. I can't possibly go through examples of each one of them. But what you've got to think about is the strengths and the weaknesses of that, and how does it push you along that road to try and answer your questions. And of course, all of the above are iterative. One doesn't come without the other, and you're completely working them together. It is very important you understand with your experiment and your study methods what they will be, and you understand actually how they are done. And sometimes that can be complicated these days because actually there's a machine will do X or Y for you. And some people argue you don't need to know this machine needs to work, you just need to be able to sort of interpret the answer. So I think you have to have some caution in there. And of course, it's impossible to do any study without understanding what numbers you need to test your ideas, what's the power of the analysis you need, and of course you need statistics. But you need statistics before you start your study, not afterwards. So the classic thing is you get a bunch of data, you give it to a statistician, say, can you find something interesting? That's probably old age stuff. We don't want to live in that world. The other thing which is common about all studies is related purely to the idea of robustness of the system and results. And that's important. It's the R&R, as I would call it, repeatability and reliability. If you're looking at a study, you need to think about how robust that study is. For example, if I was doing it in Wales, is it actually going to be robust if I did it in Scotland or in Argentina, for example? So once you're up and running, ready to start the study, you need action. It's no good actually just saying and talking the talk. You've got to walk the walk. And for that, you're going to need enormous support and tenacity. I gave the example of the HAB Select study being almost 10 years work. That's an extreme, but you do need a lot of support and tenacity if you're going to go forward. And lastly, but not least, you must publish your data because, of course, you've used public resources, you've used patient samples, etc. It's incumbent upon you to publish that so society can benefit. So there are lots of information available out there so you can actually have a look and get some feel for how to design a study. So this is Jack Cohen's book. Jack Cohen was a reproductive biologist, and he controversially put the title of Stop Working and Start Thinking. And I think the way that he was trying to get at it was to say, start thinking about the problems you're trying to address. Don't just carry on regardless type. But this book is actually very good. It's readable. It's got lots of reproductive biology examples in it. And it talks about experimental design, talks about statistics and why to do statistics, for example, and how to do them, and why and how to design a study. And I'll be using some of the examples from that in my presentation. The other book that I think is really important is actually from the Nobel Prize winner, Daniel Kahneman. And this is a description of how the mind actually works. It's called Thinking Fast and Slow. And it really deals with biases. There's a YouTube video actually highlighted here where you can look at, and he actually gives examples from the book. And bias is really important to deal with in your study and your experiments. We all come with various biases, and we can carry that bias into our experiment, and we've got to be really careful to understand what that bias is and, of course, control for it. And I think this is a great book to actually give you really everyday examples about how the brain thinks and how it can really mislead you, particularly when you're trying to do experiments. There are many other resources available. Of course, you are not alone. In academic authorship programs run by ESHRE, there are probably 40 or 50 lectures given by authors, experimentalists, statisticians, etc., which talk about all aspects of study design and how you actually plan a study, how you execute it, what's a meta-analysis, etc. And it's a fantastic educational resource. In addition to that, journals have resources. So, for example, there's a Researcher Academy from Elsevier, which talks a lot about how you actually plan a research study, how you manage your data. And other journals give you real clear guidance on how how you choose your methods, what methods to choose. This one, an example comes from PLOS, for example. So there are many, many resources available to you, and I suggest you arm yourself very strongly with getting this information prior to executing your analysis. So let's go into a little bit more detail how we do this study. First and foremost, you've got to have some sort of a question. And that's important because it's also what type of question you're interested in. What question do you really want to answer? For example, are you interested in the minutiae of detail about something? Is that really what gets you excited? For example, if you're looking at ion channels in sperm, I worked with somebody who really was only interested in the minutiae of how Caspar worked. Had no real interest why Caspar was there in the first place, or why it was only Caspar, or the evolution of Caspar, or anything. It was just exactly how it works. He was obsessed by it. He was not actually interested in anything else. That was his real brain working, and how he was interested in actually addressing the types of questions he wanted. Other people are not so interested in the hows of something. They're much more interested in the whys of something. And Jack gives the example of the porcupine, so I'll leave you read that in the book. But basically, there are different types of questions. You've got to get a real feel for what really interests you, what type of question that is, and how you can then best execute that question. And it's important you understand that it's got to excite you in the beginning. So let's assume you've sort of got your question, you've sort of got it in your brain, you're thinking about it quite a bit. Yeah, this is what I'm really interested in. This is the type of question I want to ask. This is the question I'm going to ask. The next thing, you've got to intelligence gather. And what I mean about this is not just reading the literature. We'd expect you to do that, of course. And you can do that from traditional sources such as PubMed, from Scopus, etc. You've got to have a real feel of the literature, of where it's gone and historically, and where it's actually likely to go. So you've got to do some projections on that. But this is really only the tip of the iceberg of actually the information you need concerning your study and your question. I would strongly suggest you discuss your ideas with colleagues. And in fact, if somebody's published a study recently or some time ago, in fact, around the question you're interested in, contact them, ask them. Let me give you an example. So I was interested in globospermia, which I'll mention in coming up. And I was particularly exercised by a number of cases that we were doing in the Farah Hospital in Jordan. And Doug Carroll had published a couple of papers on globospermia. So I contacted Doug. And what it showed was actually the number of patients he'd published was a small reflection of the numbers that he actually had. And therefore his knowledge base was much larger than what was observed just from looking at the papers. And that was really informative to help us go forward with our studies. So that is an example of a tip of the iceberg. Often people have a lot more data than they actually publish. And they actually choose to sometimes publish only one aspect of their data, but they've got the other aspect there, but maybe they haven't published for whatever reason. You can ask them about that. Now, generally, people are really worried about talking about their ideas to other people. For example, they'll often say, well, they'll steal my ideas. Well, you know what? Very few people have original ideas. So I really wouldn't worry about that. And actually, occasionally, maybe they do. Maybe they do. But it's a rare event. And what you will gain from talking to somebody about your ideas is much, much, much more than you will potentially lose. So I think it's very important you discuss your ideas with other people. They can think of many other things, and that's really helpful. For example, look at grant reviews. So who's been given a grant on my subject recently, for example? And then look at the abstract and contact the people. And if they're doing exactly the same as you might want to rethink what you're going to do. Also, I'd strongly encourage you to be interdisciplinary in what you're doing. You're not trying to dilute what you know, but what you're trying to do is increase the impact and increase your knowledge base. So You've got your idea, you've got your thought process, you've done all your intelligence gathering. The next bit is really get some sort of working hypothesis. There's lots of information and advice on how to do this, but you do need a hypothesis. And actually generating one is easy. In fact, it isn't that easy. You can generate a number of them because you're probably trying to test different things. So if we take the HAB Select study, for example, published in the Lancet, they were specifically interested in testing a hypothesis that PICSI improves full term greater than 37 weeks gestational age live birth rates compared to ICSI. That was their specific hypothesis they were testing. So you've got to come up with a hypothesis that can be tested. And that's a critical thing to do. Don't just come up with an aim. Come up with a hypothesis because that really will help you. And in fact, it's essential to doing that, particularly if you're doing any type of study towards a degree. If you're doing a PhD, for example, the first thing the external examiner will ask you is, what is your hypothesis? And if you look blank, you're in trouble. So what about general experimental bits? Well, the context of your experiment is very important. You know, how much have you actually dissected out to look at the context of your experiment? If you're, for example, looking at bird tables in Scotland, you know, are you looking at certain areas of Scotland? You know, is it one type of bird table? You know, what is the context of your experiment? Because actually that is an important aspect of how you manage to come at it. People get confused between parameters and variables. Basically, parameters are what you can potentially control for. So, for example, if you're looking at male infertility and you want to control for female factors, then, for example, you could test egg donation as an example. Variables, of course, are things that you modulate in your experiments. And of course, that's the advantage because what you're going to do is modulate these variables, test them either one by one, and then maybe do some add-back experiments. And of course, controls are essential because what you need from your controls is to tell you that your experimental result can be valid. So you need your negative and your positive controls to be run to say, okay, they work, therefore I can accept the result of my experiment. Whether I like the result of my experiment is a different thing. But of course, as long as your controls are run appropriately, you should be actually okay. When we're talking about statistics and design for the experiments, then we use for our students, for example, two commentaries from the EMBO journal, which are very helpful in guiding us for how to design an experiment and the statistics relevant to those designs, for example. Power, how you do a power analysis. Power analysis is very important. So if you're trying to look with treatment A versus treatment B, and you're using, for example, clinical pregnancy rates in vivo as an outcome, you're going to need a lot of patience. You're going to need to estimate what your pregnancy rates are, for example, in each group. And then, of course, you're going to have to go through a power analysis. And there's various ways of doing that to actually test what would be a clinically significant difference between those, or in fact, a significant difference between those. But you're likely to need a large number of patients. And it's important you know that before you start. And that's where a power analysis, for example, comes in. These two papers talk a lot about technical replicates and biological replicates, for example. And what this is here is some people actually get confused between these two, but it's very important you understand how good your technical replicates are and what your biological replicates are. And when it comes to replicates, it's very important also to understand that your research needs to be reproducible. And quite recently, probably over the last 5 to 7 years, there's been much discussion about reproducibility in life sciences and in medical sciences, because the worry is that actually a study done under very specific conditions may not actually be reproducible, either under those conditions in another laboratory, or in fact not reproducible at all. Now, I'm ignoring here, of course, scientific fraud where somebody would make up the results, but basically your studies have got to be reproducible. And if you're interested in this subject, go to the Nature of the Science website and they'll give you a lot of information about reproducibility. There's a lot of information in the journals about what methods and the quality of the methods to use. So for example, this comes from the British Journal of Pharmacology. So they're suggesting if you're going to do an in vitro study, as an example, drug A and the effect on cell B, for example, you need a certain experimental design and analysis and N numbers. And they're very specific about this. That will help guide you, of course, in doing your experiment, you know? So actually look at the journals and get a feel for what they would expect for methods. Some other journals will have specific methods that you need to follow, specific systems of methods that you need to follow, and protocols. In some, they will need, for example, a detailed protocol to actually undertake your study. So let's have a look at some examples of types of studies, and let's look at some reproductive biology in those. Well, of course, there is a case-controlled study, an observational study in that way, depending on how you want to define it. These are, there's advantages and disadvantages to all these type of studies. There's an association study. There's retrospective studies where you go into your clinical database, for example, and find out what's happening there. Case-controlled studies. There's detailed experimental studies, and the experimental design could be a plethora of different examples. You can run a little pilot, which is also very helpful, certainly to reject your ideas pretty quickly. If you think sperm always turn left, and first thing you do, look down a microscope and they turn right, then clearly you need to rethink. So these experimental design can be really quite complex. Result reversal, which is basically talking about restoring function afterwards. Competition type experiments. This is often done actually with, as an example here, with sperm selection, when they're trying to sort of push the system as hard as they can. There are, of course, clinical trials, and there are a plethora of different designs for clinical trials. Randomized clinical trials, of course, being one of them. And other studies now are, of course, systematic reviews, and often they can include primary data analysis, or in fact, network analysis. So let's look through a couple of examples on this. First of all, let's look at globozoospermia as a case study. This is an important case study because actually it was first described in 1971 as an observation, as a case study. And on its own, I guess, it could have got lost in the literature. But basically, it showed that actually these cells appeared to be quite frequent in infertile men. Then subsequent studies showed that the cells failed to fertilize, but ICSI worked. And now the gene's been identified in 2011. And now we can understand, if we can identify the genes, is there a bit prognosis comes from those? For example, if one man has got a particular gene versus another man, have they got better prognosis and diagnosis? And also we could then think about artificial oocyte activation for these men. So actually, resulting from an original case study, we've understood a great deal from, for example, globozoospermia. So although case studies are rather rare these days, I guess, and the journals don't really like them, there's still a lot of power to an appropriate case study. There are, of course, association studies. This comes from, for example, the Beckwith-Wiedemann experiment, effectively, or association, that we did many years ago when I was effectively talking to somebody in a coffee room. He was an Everton supporter. I won't hold that against him. And I support, of course, Aston Villa. So I was just chatting to him, and he was a geneticist. He was an expert, Eamon Mayer, in Beckwith-Wiedemann syndrome. And there'd been a case report of an Angelman syndrome in ICSI patients. The idea was that maybe there's an increase in imprinting disorders after ICSI. So after talking to Eamon for over a couple of cups of coffee, he came up with, well, actually, we could look at our databases and see if the incidence of ART in our Beckwith-Wiedemann syndrome patients. And then we can compare it then to the general population for ART. And if there is actually a significant difference between those, maybe there is an association. Now, this paper was published in 2003, and it's the highest cited paper I've ever been involved with and written. So it's clearly created quite a lot of an impact, but it's an association study. It's not a causality study. It's just how many patients with Beckwith-Wiedemann's AD-ART, and then looking at the general population. So it's an association, but actually it kicks off a whole idea and concept. The next type of study that is very common performed is a clinical trial. Now, clinical trials are actually very challenging to do well, particularly in assisted conception. There are a large number of complexities to delivering a clinical trial. And as a consequence, I'd strongly recommend that if you're thinking of performing one of these, that you contact your local clinical trials unit. Most hospitals, universities, research organizations have a local clinical trials unit, and they basically perform clinical trials on a professional basis. They will give you the ins and outs and the A to Z of how to perform a clinical trial. So I'd strongly recommend you talk to them. There are a large number of things to consider, particularly for regard to the design, what controls are going to be used, what endpoints are going to be used. So for example, if it's in a laboratory, are you going to use the endpoints, for example, of embryo grading? Are you going to look at implantation? How are you going to randomize your patients? How are you going to use particular types of controls? Is it going to be a randomized control trial, etc. So I would strongly recommend contacting your clinical trials unit. And what you see actually from clinical trials in ART is they're actually pretty rare. So if you look, for example, on the National Institute of Health Research in the UK, the reproductive health aspect of that organization, you'll see that they spent in 2017 to 18 $21 million on research studies into reproductive health. So there's a very, very active clinical research network in reproductive health. However, very few of those are actually done on assisted conception. So although there were 95,000 patients recruited to studies in that year, very few actually were for assisted conception. It is very challenging to do studies in this arena. So here we have one example of a very recent clinical trial in assisted conception. And this is published by David Miller and colleagues in The Lancet, and it's examining HAB-Select, which is looking at a sperm selection technique for ICSI. One of the key parts of design of this study and the pushing forward of the study is the time interval. So for example, David, following getting the idea for this study, would have needed to design the experiment, get the funding, do the study, and then publish a paper. And in total, this would have taken between 8 and 10 years from inception to actually delivering the paper at the end. So these are a complicated undertaking and not to be thought of lightly. So I would re-emphasize that if you're thinking of doing a clinical trial, contact your local trial units. We need many, many, many more trials in assisted conception, but they're not actually easy to do. And this is an example of a very high quality, well-performed study. There are, of course, other types of studies, and an example here is a meta-analysis study, which basically looked at a systematic reviews related to, again, this is sperm selection as an example. So this is where all of the data is examined systematically, and this is from a Cochrane review example. And what we have here is a conclusion related to the fact that further high-quality studies are required. And often this is the case in this particular type of analysis, because that's part of the purpose of doing it in the first place, is that we're not completely clear about the evidence, and a meta-analysis helps us analyze and put forward and drive that evidence. We can take the meta-analysis one step further, and this is a network meta-analysis. I couldn't find a detailed example of reproductive medicine, but actually this comes from the work of John Ioannidis, and it's looking at antidepressants. And this puts together all of the data, I think on about 100,000 data parts, to look at the differences in the antidepressants currently available and historically available and how effective they are. And this is incredibly powerful data particularly for looking at clinical evidence related to effectiveness. I'll put only one small note of caution in doing any type of study, and that's the power of new technology. The example I would use here is that actually for a long time in fertilization studies, Adams and Adam receptors were said to be essential for sperm egg binding and fusion, and there were hundreds of experiments demonstrating this, but they were never completely that clear. They were either using peptides or antibodies, and the experiments were always a little bit dirty. So when knockout technology came available, then what we were able to do is then knock out these particular sort of key receptors and see whether fertilization occurred. And of course, lo and behold, Masura Okabe, who's published lots on this area, knocked out a lot of these ADAM receptors, for example, and shown there were no importance whatsoever. And in fact, if one paper you should read related to this type of experiments, it's this one from Okabe, because actually this really puts forward the whole idea of powerful methods and how they can answer questions. So what particular challenges occur in an ART environment if we're actually trying to do research studies? Well, there are substantial challenges. I think first and foremost is the fact that actually a lot of ART is actually private. So therefore, there is somewhat a slight divorce between academia and the private medicine sector. And I think historically that's not been the case. That's in fact, of course, how IVF came in the first place, was from the collaboration between private medicine and academia. But this divorce is probably no help in the ART environment in general, because training, opportunities, the skill level of people is probably not ideal without a scientific background. And of course, there is a numbers issue. Many clinics actually have relatively low numbers in their labs and in their clinics. So doing studies, particularly large studies, is going to be very challenging for those individual clinics. Our interdisciplinary network in ART is probably less sophisticated to other particular disciplines. And I don't think this helps us that much. And in general, the number of clinical studies we do, particularly in ART, is really, really, really low. Hans Evers used to say that this was a real Achilles heel of reproductive medicine. We have huge constraints on our material, and of course, this is just nothing we can't do anything about it. For example, if we want to study fertilization in humans, you've got to study IVF, but of course, these events are relatively rare in individual clinics. If you want to study human embryos, you have to study human embryos. You can't study any other embryos. And of course, getting human embryos for research is really difficult. If you want to study metaphase 2 eggs, it's really difficult to get donation of metaphase 2 eggs. And within that, there's a significant ethical, legal, and regulatory framework. And of course, that can be prohibitive. For example, embryo research could be banned in your country. Therefore, you're not going to be able to do it. So there are challenges in the ART environment, many of which actually we can't easily get round, and we absolutely have to deal with head on. However, the other side to the coin, I think, is much more powerful. I think, but I guess I'm biased because I'm a reproductive biologist, that actually this is the only study worthwhile, the only subject worthwhile studying on the planet, to be frank. Basically, if we can't reproduce the next generation, we're not going to need to worry about global warming or anything like that, because actually we're not going to be here. Nobody's going to be here. So it's critical to evolution that we have reproduction. It's fundamental to people in society. Having children is anybody who works in ART is completely obvious at that point. And of course, from an experimental point of view, of course, it's difficult to get human gametes and embryos, but actually the big advantage, you don't need to justify you've got an animal model, because actually you've got them, you've got humans. You don't need to justify anything from that point of view. People who work on animals constantly have to write, oh yeah, but my model's relevant for X, Y, and Z. And often, of course, that's rubbish, to be blunt. So actually, that's a massive advantage, huge advantage that we probably don't think about initially. Our field is rapidly growing. It's really dynamic and exciting. And that is not the case for all other fields. So that's a big advantage of assisted conception. And we have got exceptional translational speed. And this is what I think the involvement in industry is really powerful. If you come up with a good idea tomorrow and actually it's ethical and reasonable and it works, you can get it translated really, really quickly. And that is really important for trying to help patients and society. And I think public awareness in our discipline is massive. Interest and support is very high. We don't have to sell assisted conception. We don't have to sell reproductive medicine. I work in a department where they work on much less interesting subjects, and they have to work really hard to try and sell their subject. We don't have to do that, and that is a massive advantage. So I will lead you now with a summary of what I've tried to go through, ladies and gentlemen. I think studies are essential to what we actually do. We should be doing studies pretty well much all the time, but we should be thinking very clearly about how we design and what questions we're asking for those. I've gone through the basics for a study, the types of study, and given assisted conception examples, hopefully. And what you should do next is have a good think about what questions you can then ask, and then talk to people and say, okay, well, I need to do a study on this because actually we don't know the answer to it, despite the fact that we think we do. Thank you very much.